Hello everyone. How are you doing? Are you doing well? Nice, love it. Thank you very much. I love the PyData conference. A lot of energy, a lot of very interesting talks and people. My colleagues over there love going to the PyData as well. So this time, this talk will be about processing medical images at scale on the cloud. I know you know how to read, but I have to do it anyway. Hello, my name is Guillaume. I'm from Paris. My title is software and data engineer. It's very important for me to cross both because data engineering and software engineering, they throw rocks at me. The company is Twig, like my colleagues. So they told you enough about that. Shameless publicity again. We're a service company. If you want to build things that are incredible, come work with us. So I love Python, machine learning, and this is why I'm also going to work with Go in about a month. But still, I've worked a lot with Python. I like to think that I know how it works, and this is why I'm presenting today. We have an amazing blog, which is written by engineers for engineers. So if you're a nerd like us who likes programming languages, compilers, build at scale, data science, machine learning, cloud, whatever, go check out the blog. It's really, really good. Okay, so today, what are we going to talk about? We're going to talk about whole slide images and how they can help against cancer. We are going to look at challenges that come when we want to train neural networks on such images. We are going to see how we can empower machine learning engineers like researchers when working with this kind of data. So it's about medical images, but a lot of stuff here applies when you work generally with large data that is not tabular. Oncology. Is there any clinician or anyone working in oncology or close to oncology in the room? Okay, very good thing. So you're on the second row. So if I say anything stupid, you take your phone, you throw it at me, okay? Run in your face. And then you can correct me in Q&A, please. But anyways, oncology is the study, treatment, diagnosis, and prevention of cancer. I am a software engineer, data engineer. I don't know much about it, so I will keep it very simple and not pretend that I'm an expert. Cancer is bad. And diagnosis is complex. That's basically all I can say faithfully. Making a diagnosis. Usually, a clinician would sample bloods, do radiography, imaging, to see if you have cancer, if it's spread, and what type of cancer that is. And one of the important steps is biopsy. Biopsy is the sampling of cells to study the type of cancer you have. So for instance, let's say I have melanoma in my hand, I would scrap some bits of it to be able to analyze it under a microscope, right? Different types of biopsy, but anyways, cells. That's all that matters here. And then you prepare slides. And a slide is something like that. So a slide is a piece of glass, and you put the cells, and you do some treatments on that, and you end up with something you can study. So you prepare those slides, and then you capture zoomed images of them. This is what we call whole slides, images. Right? It looks a bit like that. This is one of the test files from a library. We're going to talk about that later. These files are super, super big, right? Here, it's not 4 cells. It's 4 pieces of tissue. And then you can't really see on this screen, but it's plenty of cells. We talk about MPP. MPP is micrometer per pixel. So the more zoomed in you are, the lowest the MPP, right? It's only a few micrometers by pixel. This doesn't suit all the usages because we do a very zoomed in image. And so to be able to, for instance, just have a look at the general image, you would need downsampled images. And we usually store pyramid of images then. So the 40 times zoomed in would be, for instance, for this example, 200,000 pixel and 150,000, right? That's a lot of pixel. And then you would downsample it by factor of 2, so you would have zoom times 20, right? And this is maybe more manageable for some other systems. But when you want to do machine learning, for instance, you probably want to use the more zoomed in. And when you want to just check the image, have a thumbnail, you probably want the less zoomed in. So it's a pyramid. And also these images are too big most of the time to process. So you would want to, for instance, process in a grid and just look at one cell of this grid, check the cells in them, do some processing, etc., etc. And also you can see there's a lot of cells in this grid that don't even have any cell in them, so you don't even want to process all the grid. But that's a topic for later. So you want to make a diagnosis. So you did capture those whole slide images, and now you want to detect cancerous cells. And this is where it comes, machine learning to the rescue. So this is a picture from the YOLO website. Or you can do segmentation as well, right? Who does machine learning on images here? So I know. Okay, I thought there would be more people. Okay. Maybe it's old now. Maybe Gen AI is the thing. I don't know. So goals here for machine learning in oncology will be to detect cancer earlier and increase chances of survival. Yay. To help clinicians spend less time on tedious tasks, because you've seen there were a lot of cells, right? So they maybe already know where to look at, have an idea, but they do spend a lot of time on that. And even if you give like regions where it's likely, but you're not sure that there's cancerous cells, but you can exclude all the places where you're sure there isn't, that's a huge amount of time you've gained, right? And then you can focus on other things because they have a lot of things to care about. So why going into the cloud? Then you ask me, this is in the title of the talk. Well, this is one of the part where you can throw rocks at me. So hospitals, they use on-premise storage system most of the time. For instance, for DICOM, they use PACS. Oh yeah, I see someone nodding. You know what I'm talking about. It's decadal, not really flexible. It was designed at a time where compute and network was something else. And so you have limited on-premise compute. I want to insist on that as well. Why the cloud then? Because you would have flexible storage and powerful on-demand compute, really. This is arguments I've seen in that field. So what is the plan? The plan is to ingest those images, those files, to cloud storage. In this case, we were using Azure, but you could use different cloud storage. Then train neural networks for detection, segmentation, and then you can use them using a deployed inference service, right? This is basically 101. So then the first challenge, how do you read these files? Well, it's not like a PNG that you just double click on it and Windows Explorer can just show you in the file viewer, right? It's a bit more complex. First thing is that you have to go from these files, the way it's serialized inside, encoded in the file, to rasters, right? When you do, for instance, convolutional networks, you need raster. What is a raster? It's very simple. It's a grid of pixels, right? And you need to do a convolution, you need a grid of pixels, because that's what machine learning does. Who doesn't know about CNN here? Nobody, great, okay. So you know what I'm talking about. First order challenges are the size of the files. As I said, a big amount of pixels at the lowest level. It can be up to 10 gigabytes. I've seen files of 10 gigabytes. Unconventional formats. So the most conventional format you could find there would be TIFF, which was designed by Adobe, I think. And then you have SBS, Mirax, and DICOM. And something like Mirax is proprietary, so you also have to reverse engineer it. Of course, you don't have to do it yourself. The community has already done that, but it's a topic to consider. And yeah, so you don't want to have one big raster. You also want to process in regions and or patches. So the library we mainly use in that field is OpenSlide in Python. It's open source, which is great. And it reads most of the whole slide images formats. Yay. It's implemented in C. I love that. It's fast, it's efficient, and it has Python bindings, which is really easy to use. This is the kind of code you can end up with. So you just put the file name or the path to the file that you want to open. And you can access the metadata, for instance, how many levels you have. Levels are different zooms, right? Dimensions at the lowest level, so the biggest image. The downsample factor between levels. And you can specify XY level with height, and you get a proper image you can use a raster using this read region method. So this is really cool. We're almost there, right? Well, it's implemented in C. And so it reads from standard C file handlers, right? And in the Cloud Rail, we would have rather wanted to feed bytes or IO bytes. Because reading a C file handler like that, we need the file to be on the machine. We can't read directly from the blob storage. So what do we do? Solution one, you copy the file. You just download it. It's really good because it's simple. You don't need any setup. And if you have to read the entire image, anyway, you were going to read all the bytes. So might as well just copy them entirely. The problem with that is that it's inefficient if you want to read only specific regions. And as I've told, you don't really want to read the entire image most of the time. So second solution is Fuse. Fuse is Linux user space file system framework. And basically what it does is it will allow you, using a kernel module, user space library, which is libfuse, or the mount utility fuse amount, to be able to use the files in the cloud just like if they were mounted on your hard drive, right, in your file system. This is like if you're plugging the cloud storage just like a USB key. So that's really awesome. The library to do that on Azure is blobfuse2. And it will communicate through the REST API, so on the network. It has a stream mode, which at the time I was working on that was experimental. Now it's more established. It's good because the old way it used to work was it would download the entire file first and then read the bytes, which was not really the efficiency we were looking for. But now with the stream mode, you can only read the bytes in the cloud object that you need, which means that you only transfer so much. This is how it looks like on a Linux. So you would mount using the blobfuse2 commands, and then you can just use the standard file operations like going to a folder and creating folders and putting information that it would do the read, the write, whatever you need. So that's great. The pro is transparent. You don't have to change your Python code. Let's be real. This is the kind of thing we want. The problem with that is that it's more effort to set up, right? It's a bit more of a DevOps thing. You need to install, you need to configure, and also when it's Dockerized, you need more capabilities. And well, for instance, if you use managed Kubernetes, you don't necessarily have the ability to use these capabilities, so you can actually do that. And also, who has a Mac here? Okay. A good proportion of people. I don't actually, but yeah, it doesn't work on Mac, which is a problem for data scientists and companies that have data scientists working on Mac, not to be underestimated. Also, the cache configuration was hard to get right. I don't know about it anymore. The third solution is a library called Tisflied that is developed by Andreas Pohlmann. I've had the pleasure to work with him. He's really a great guy. Go check out the repository. It's a drop-in replacement for OpenSlide. So you can see that we import it, and you can use it to have a slide object, just like you would with OpenSlide. And then this object gives you the same methods and properties as OpenSlide. So this get thumbnail is also a method you have on OpenSlide. But as you can see, I didn't specify a path to a file, but a file handler using fspec. Who knows about fspec here? Doesn't know about fspec? More people. I highly recommend if you want to do more like MLOps, you have to check out fspec. It's almost standard in Python libraries. So it will give you like a standard file handler in Python, so not in C, for operation like read, write, and operation on file systems. And it works with clouds. Yeah, so with TipFlight, you will only read the relevant bytes, as FSPec does. The good thing about it, you have no additional system configuration, and it's based on FSPec, which is pretty standard. The problem with that, it supports less format than OpenSlide. And also, it's not as performant as OpenSlide, of course, partly because of FSPec itself, partly because of like the library itself. But it's pretty good as already. So here is, for instance, the kind of data set you would implement for Torch. So this is going to be the example. And you would specify, for instance, in this example, the list of URLs of objects that are whole slide images files in your cloud storage. You can open them, and then you can yield regions. So this example leaves the entire region at level 0. This is not what we want, right? We want to have patches. Otherwise, it crashes, because our whole slide images are too big, as I've said many times. So we cut into patches. The grid, once again, this is going to be our first approximation. But you could want to have different strategies, because maybe you have cancerous cells that are hidden at the edge, right? So maybe you want to stride differently. So this is, for instance, an implementation that discards the logic about how you make patches. Let's consider that you can specify a specification for a patch with a level X, Y, and size. And then this dictionary would be the URL to the file in the cloud storage, and the values will be a list of the patches you want from this image. I'm leaving the logic up to you. You can use detection beforehand. You can do some pre-processing, whatever you want. The interesting part for me is what's in the iteration part, where we will iterate on these specs to generate specific regions. But it's slow. What we found was that doing that, it would take too much time to read the data and prepare the patches, to prepare the training batch, right? To stabilize the gradient when you do training of models. Theoretically, you want a batch size of 1. If you follow the theoretical guys, we know it doesn't really work, so you do a batch to stabilize the gradients. And what happened was that the time it took to do a single training step in the loop was less than the time it took to load the patches, to have a batch. And we want the inverse. We want it to be faster, because otherwise there's GPU time we don't use. And this is where parallelism comes to the rescue. We just make it parallel, so we divide by the parallelism we have to make it faster. But the problem was that it was not enough for us. I think at the time I did the computation with benchmark, we needed parallelism of 90 or something like that. We had good GPUs. And so this is more than one machine can handle, and the solution to that is distribution, of course. I heard it, so well done. So the idea is that the user is going to be working on their laptop, and they will connect to a cluster, and the cluster will be able to do more parallelism. So you need distribution. In my experience, this is not what data scientists / machine learning researchers want to focus on. So here's come the part where we have a dream, where we just have code like that. We define a data loader. We don't necessarily implement it ourselves. We have a trainer. If you don't know PyTorch Lightning, I really recommend it. And then you do like, just run the experiment for me, and I don't really care about how it works and the infrastructure and all that. The way we implemented that was to use Ray. So Ray is a library that it comes from reinforcement learning, I believe. There was RLlib that was a great library to do training for reinforcement learning algorithms, and like a gym with that. And it was doing a lot of distribution, but then it became more general. Who uses Ray in the audience? Oh, quite a few people, okay. So, and it comes with a set of Helm charts that you can deploy to Kubernetes as well. So for anybody who doesn't know about Kubernetes, you should have a look at that. It's really, really interesting. But basically what it does is it will orchestrate Docker containers in virtual machines, right? In a fleet of machines. And so you could have easily auto-scaling, you can have health check, you can replace the containers when they die, you have a bunch of really cool stuff with that, and monitoring, etc., etc. And the way to deploy this kind of setup, usually you can do a lot of manual things yourself, but we know it doesn't really work. So there's a bunch of libraries to do that. There's Customize, there's etc., etc. But there's one that's called Helm charts, and Helm charts are really easy to use. Ray has a project that's called KubeRay that provides such Helm charts so that it's really, really easy to create clusters. It's effortless to create clusters, honestly, if you know how to use a terminal. It's kind of like Databricks, but without a UI. So how does it work? You import Ray in the script where you're gonna do the data processing / training of the model. And this init function, you can use it to connect the entire Ray thing that you're running to a cluster. And then you can have this kind of implementation where what you do is you define a Ray data set. So here you can do that from a pandas data frame. The pandas data frame is just going to be one column URL of the file and one specification of a patch. And then you have many of them. And you have a data frame. You can turn that into a Ray data set that is going to be distributed. Here I'm doing a bit of repetition, but don't mind this for now. And then we do this map of read patch. This map is lazy. It's not executed when I'm just getting the object. It will be executed on whatever I need at every iteration when I generate a new batch. And it's going to be distributed. So then I have this dataset.toTorch method, and it effortlessly converts to something I can use with PyTorch in my training loop. You can see the batch size specified, for instance. So quite easy to use. And then, for instance, we were using a cluster with one head node and 8 workers. And the good thing is the head pod was doing the training of the model, so it had to use GPU. But we didn't need GPU for the data processing. And so we can spawn as many of them as we want. It's fairly cheap. And then we can reach the great parallelism that we needed. To submit a job, you have to connect to the cluster from your laptop, however you want. Here is just an example using port forward directly from the Cube CLI. And then you can use the Ray CLI, which is what interests me here, where you submit a job and you specify the Python module in your current working directory that you want to work with. What's really amazing is that Ray takes care of the whole environment thing so that you can work with a module instead of a script, so which is, I think, much more convenient than just sending a script. You can also specify additional extra dependencies if your experiment needs more Python packages. You have a bunch of options like that. And then you run whatever you want. So it's general purpose. If you want to do local development, just don't specify any cluster to connect to. Just do ray.init, well, value of none then. And it will run locally for your development environment, right? So you don't need a cluster to set up a cluster locally to start developing your data processing. So what did we talk about? Whole slide images are important for the future of digital pathology. And machine learning on non-tabular big data is not trivial. But we can help people focus on the actual research by developing the infrastructure, architecture, and tools so that they can really focus on just training algorithm and their actual job. Thank you very much. It's been a pleasure attending this conference, and yeah.
Thank you. Questions?
Apparently, I was very clear.
Yes. Well, let's thank our speaker again. Ah, no, there is a...
You can't escape the mic. Yes. There's no way.
It's recorded. Yeah, just you mentioned the different file format, and having working in biology, I know they are a pain to work with because all of them are proprietary and they're really a major pain. Were you able to circumvent that, or did you still had issue, or at least did you add some uniformity in the formats you were working with? Thank you.
That's a very good question, and I think it's not just about biomedical images. It's also generally about big systems like that. I know there's the same kind of problems with spatial images, I think, from satellites. It's very proprietary, maybe not in the recent versions. I mean, more recent gear. But yeah, it has been a problem. And for instance, I mentioned TIFF. TIFF is a very good standard because it can hold a lot of metadata, and then the image is serialized, not like a PNG with like every row, but you have tiles, and each tile is like a PNG. And so you read only the contiguous bytes of the region you're interested in. And that's really good. That's fairly open. That's known. And this is why people just don't use this format. Honestly, I don't know why. We had to work with Myrex, on the other hand, and TIFF slide does not implement yet Myrex. And Myrex, actually, it's from a system that does it captures the whole slide image by having a microscope that will shift with the motor. And so it's not really precise, and so you have these overlaps. And it's a pain to. I had to implement a custom library to handle these images, and yeah, it was really complex. So, yeah, the stitch. So in some Myrex file, you do have the information to stitch, and in some files you don't. I don't know why. Maybe older machines. So yeah, I would say in this kind of field, you have to consider this kind of effort that's non-neglectable. Yeah. Does it answer the question? Thank you.
So let's thank our speaker again.
Thank you so much.